Functional differences in the nucleus accumbens between adolescent nicotine smokers versus non-smokers using fMRI. Nicotine is one of the most addictive substances in our current society due to its easy availability, influence over our neurobiological functions, and strong cognitive reinforcement as effects are nearly instantaneous. Cigarette consumption has been in a long-term declining trend for many years with increased awareness on its negative health effects. However, electronic cigarettes production and consumption has soared, with 50% of Australian individuals smoking daily, a 42% increase from 2019. It was originally invented to aid quitting nicotine, but it's now commonly used as a first nicotine experience in adolescents, with 3.5 to 9.3 of 14 to 24-year-olds smoking daily. On average, a vape contains the same nicotine content as 50 cigarettes, and 80% of individuals relapsed within 1 month after quitting. This displays its immense addiction risk. Smoking is known to harm almost every organ in the body. It kills more than 8 million people yearly, either directly or from increasing vulnerability to other health issues, including cancer, heart, and lung disease. It's also supported to have lifelong effects not only on health, but increased risk of other substance use disorders, gambling disorder, poor impulse control, and detrimental effects on brain development, especially in adolescence as the frontal lobe hasn't fully matured. Adolescent functioning and development are characterized by an imbalance between the limbic system, which aids regulation of emotion and behavior, and the dopaminergic mesolimbic system, otherwise known as DMS, which mediates the cognitive and psychological processes of rewards. The DMS is a circuit between the ventral tegmental area and the nucleus accumbens, where the dopaminergic neurons project to the stratum, prefrontal cortex, amygdala, hippocampus, and other limbic system structures. The reward circuit promotes the release of dopamine when the brain associates a stimulus or behavior with a pleasurable or desirable outcome. This includes natural reinforcing stimuli from everyday experiences, such as good food or sex. However, after repeated exposure to addictive substances such as nicotine, it causes a dysfunction in the dopaminergic mesolimbic system as it's a strong unnatural reinforcer with direct agonist properties that releases significant amounts of dopamine with every use. This results in an overall decrease in the reward circuit sensitivity and long-lasting decreases in the D2 dopamine receptor. This results in less dopamine being released during naturally reinforcing stimuli and more released during nicotine use as it's amplified the body's natural threshold. This continues until tolerance occurs when the individual neurobiologically needs to increase smoking intensity and frequency to attain the previous level of nicotine effect and dopamine release. The nucleus accumbens is a primary focus as it's a major substrate of the DMS involved in reward processing and incentive-based learning. It's strongly associated with initial and continual nicotine use. Nicotine-related cues such as images of smoking or vaping are supported to increase dopamine release by themselves as the brain associates the stimuli with the behavior of smoking. When adolescents were shown pleasurable images of food versus control, the pleasurable images showed reduced activation in the insular and inferior frontal regions in smokers compared to non-smokers. When they were done assessing smoking cues versus neutral cues, the smoking cues were associated with high activation in the DMS compared to non-smokers. This pattern continued into adulthood when nicotine smokers showed significantly higher activation in the DMS to smoking-related cues. The rationale for this experiment is the gap in the literature on fMRI activation studies between pleasurable images and nicotine cues. Also, it's important to understand nicotine's early DMS effects in adolescence to investigate potential diffuse lifetime effects, and we need investigation on the magnitude of reduced dopaminergic activity to naturally reinforcing stimuli, as it could have clinical implications to include it in nicotine addiction treatment by focusing on cognitively increasing rewarding associations to normally rewarding stimuli. And if the effects are significant, it could increase the empirical basis of electronic cigarettes' neurobiological effects and increase awareness for prevention and early intervention to reduce the current astronomical healthcare burden, economic costs, and addiction rates. The aim of the experiment is to investigate the different functional activity in the DMS using a fMRI bold contrast between adolescent nicotine smokers and non-smokers when presented with natural reinforcing and nicotine cue images. It's hypothesized that the adolescent nicotine smokers will have decreased activity when presented with the pleasurable stimulus and increased activity when presented with the nicotine cue images. There will be a total of 40 participants, 18 reported smoking daily and 22 reported never smoking. All participants completed a confidential questionnaire about their nicotine smoking behavior, their demographic information, their exclusion criteria questionnaire, an MRI safety questionnaire including their height and weight, informed written consent from the participants and their parent or guardian. All research procedures had to be approved by Monash University Ethics Council. And then prior to imaging acquisition, process measures for compliance to protocol were done and behavioral procedures on MRI safety were outlined. A 3T MRI scanner and Monash biomedical imaging will be used. For the pre-processing steps, a T1 weighted structural image will be acquisitioned for localization. The parameters are as followed. A field map image will be acquired for fMRI registration to a template brain to localize the region of interest of the nucleus accumbens and for distortion correction and pre-processing. And an fMRI will be done to display directional blood flow corresponding to blood oxygen metabolism associated with increased regional activation. And the parameters are as following. For the total duration, it will be 9 hours and 28 minutes for all imaging. The stimulus method will include a block design during the fMRI scans to detect activation in the hemodynamic response to subtract from the control baseline. Each fMRI acquisition will begin with the presentation of a fixation cross for 18 seconds and then a stimulus block for 18 seconds. This will repeat back and forth 3 times for each fMRI scan. The first one will be based on the first condition, which will be pleasurable images, including stock photos of good food, pleasant landscapes, and winning a race. And the second condition will be with nicotine cues, using images of vapor, people smoking cigarettes, and etc. The stimulus will be presented via software running inside the control room and delivered to an MRI-compatible screen inside the room. The general linear model will be used for analysis of individual fMRI datasets, with the explanatory variables including the presentations of pleasurable or nicotine cue images. Processing steps include the index of alignment to see the amount of head movement by summing all the squared differences in intensity across the coordinates. Then if it's a large sum, use the motion correction, which estimates the parameters and reslices the images to reduce the difference between voxel intensities to stop bold signal changes. Brain extraction will be done to simplify the motion correction by removing the skull voxels from the fMRI. Distortion correction by using field map images will be done to reduce geometric distortion from fMRI magnetic field inhomogeneity in the phase encoding direction. Slice timing correction will be done by using interleaved acquisitions to reduce residual effects of regional excitement and fluctuations in voxel intensity between acquisitions. High-pass filtering will be done to reduce the drift of bold signal intensities to avoid confounds. Spatial normalization will be done by registration to a template brain to average all fMRIs from participants with a high resolution and accurate localization. Spatial smoothing will be done to increase the bold image's signal-to-noise ratio. A comparison strategy will be used across all individual fMRI levels of analysis to identify graduated levels in the nucleus accumbens for intensity-dependent activations. The first level activation has the EVs of periods of presentations of pleasurable and nicotine cue images. Contrasts are below. The second level analysis uses a parametric design with between participant variants as comparison strategies by registering the images to a template brain via determining the geometric transformations to outline the position of anatomical structures across the fMRIs and the template image. This will allow averaging the results of the fMRI scan per participant for assessment of individual differences and corroboration of evidence to support the participant differences in brain activations, with the contrast below. Group difference comparison strategy will be used by comparing the adolescent smokers versus non-smokers for each first-level contrast, as seen below. This analysis will allow specific identification of differences in nucleus accumbens activity and therefore reward sensitivity in both groups for each stimuli. This will address the hypothesis as it will show if adolescent smokers have decreased activation when experiencing pleasurable stimuli and increased activation during nicotine cue stimuli compared to non-smokers. It's predicted that adolescent smokers will have less activation in the nucleus accumbens during naturally reinforcing stimuli and increased activation during nicotine-cued stimuli. It's also predicted that male smoking participants will have increased DMS sensitivity as they're believed to reach frontal lobe maturity at later development than females. This increases their risk of starting and continuing smoking nicotine with a higher likelihood of risky behaviors and reduced impulse control. It's also expected that smoking participants who have been smoking longer, typically the older participants, have a stronger nuclear accumbens activation due to nicotine cue stimuli than the newer or younger participants. Thank you for listening, and these are my references.